This is Shetland Sheep Genetics, a high-level overview. My name is Jennifer Johnson with Whispering Pines Farm, and to find us on the web, just type soft Shetland wool in your browser window and you'll find us. So why are Shetland Sheep Genetics important? Aside from the benefit for the breed, understanding what genetics make up the traits that define the breed, as a farmer, what it does is it allows us to have control and understanding of what our fleeces are going to look like. We're in the business of merchanting wool, and so it's important to us to be able to respond to the market demands for different colors, patterns, and fleece styles. The other reason why it's important for us as a farmer to know our genetics is that we can communicate with other breeders using standardized terms and the genetic descriptions when we're either making purchase decisions to bring stuff into our flock or helping other farmers achieve their goals by transferring animals from our farm. So we're able to communicate and clearly describe the traits that the sheep carry in their genetics. So to start the conversation, you have to understand genes. Genes are the building blocks for traits, and traits are anything that you would use to describe the animal. So some trait examples are fleece color, ability to resist parasites, and ability to keep their fineness over many years. Genes will come in pairs, and the offspring inherits one gene from each parent. In the sheep vernacular, the sire is a male parent, and we use the word dam to refer to the female parent. Genes are found in the nucleus of every cell in the body. They actually are combined to form chromosomes. They're, depending on where they are in the body and what role those cells are playing, certain genes will be turned off or not. So there's two types of traits, and in this presentation, I'm going to focus on qualitative. So qualitative traits are where there's one pair of genes only that create characteristics that we can see. So it's easier to learn genetics by focusing on qualitative traits. Quantitative traits are much more complicated. There are many more gene pairs that combine and sequence to create characteristics that are difficult or impossible to see. And also there is an environmental influence on those traits, which would be things like how much milk a ewe is able to yield, how much grease is in the wool, and their body weight at certain stages in development. So the focus on qualitative, it's just easier to communicate genes, plus that those are the ones that actually you can see, and so they, for our purposes here as wool merchants, that's really where the rubber hits the road. Another set of terms is recessive and dominant. So for each pair of genes, there can be a dominant or a recessive trait. What the sheep looks like is determined by the genes they've inherited from the parents and which genes are recessive or dominant. So for example, if you look at the trait of spotted, so not spotted is the dominant trait and spotted is recessive. So like I said before, there's a pair of genes in every sheep that determines if the sheep is spotted or not, and there's only two possibilities in spotting. The dominant not spotted is used the label capital S to identify it, and lowercase s is what's used to indicate spotting, and that's the recessive. These different genes are called alleles, so spotted and non-spotted are alleles. So there's three possibilities with spotting genetics because there's only one pair and there's only two possible alleles for spotting. So you could have homozygous for not spotting, which means that the not spotting genes are the same, so they're both not spotting. You could be heterozygous for not spotted, and that means you've got a recessive spotted and a dominant not spotted, making it heterozygous, or they're not the same. And then the other possibility is homozygous for spotted, lowercase s's. So sheep 1 and sheep 2, they're not going to have spots on their wool, and sheep 3 will have spots on her wool. Another term that's helpful to understand is called phenotype, and the phenotype is what the sheep actually looks like. So the ewe in this photograph is a spotted ewe. Her phenotype is spotted. The genotype, alternatively, is the actual combination of genes that the sheep carries in her cells. So because she's a spotted ewe, we know that her genotype is lowercase s, spot, spot. And that's the only way you would ever have a ewe with a phenotype of spotted. So we can try to predict what the sheep's going to look like as it relates in this scenario to spotting. The only thing you need is you have to know your dam and sire's genotypes. And many times that's fairly straightforward. And sometimes you don't know what they are because, you know, if you look back at their pedigree, whether they carry a allele or not is not 100% clear. So the tool that we use is called a Punnett square, and it consists of 3 sections. There's a section for the sire information, and I have that depicted here in the green background. And then the dam section is in the orange background. And what you do is you write in their 2 alleles for whatever trait we're talking about. For this one, it's spotting. So in my scenario here, I have a sire and a dam, and they both are heterozygous for not spotting. And if there's an expression we're going to use going forward, if a sheep is heterozygous for a trait, what we say is that they're carriers for the recessive trait. So the sire in this example carries spots, but he's not spotted. So his phenotype is not spotted. And the same with the dam. So then what you do is you just, working across and up and down, you put each of the letters in the corresponding square of the Punnett square there. And so the 4 quadrants in blue are what your possible outcomes are for your lamb. So for the lamb, in this case, there's 3 possible genotypes. There's heterozygous for not spotting, there's homozygous for not spotting, and there's homozygous for spotting. But there's only 2 possible phenotypes. This lamb will either look spotted or not. So what we can predict in this case is that there's a 25% chance that this lamb will be spotted. So what that means going forward is if you breed with that lamb in an effort to find out what she will produce, and it'll also help you to find out what she is. Is she homozygous or heterozygous for spotting? Is she a carrier or is she not? And the best way to find that out is to breed her to a spotted ram. If you breed her to a spotted ram and she has a spotted lamb, then you know that she's heterozygous for spots and she carries spots. Those 2 phrases mean the same thing. So what happens too frequently, to be fair, is that, and this has happened to us when we were trying to achieve more spots in our flock, is, you know, we'd breed a ewe that could have been a spot carrier, but we didn't know for sure. Bred her a couple times to a spotted ram. She would continue to have solid lambs. So we'd sell her, and then somebody would breed her to an unspotted ram that carried spots, and then she'd have spotted lambs. So, you know, you just keep trying, and depending on what the trait is, you know, we're not really focusing on spots any longer, so we're not going to do any experiments there. You know, we're looking more at other things now. So it depends on what your focus on is and what your goals are for your flock. So color is another trait that's determined by 1 pair of genes, and there's only 2 alleles in this trait also, which the 2 alleles are black or brown. Black is the dominant allele in this case, and brown is recessive. So I've put together a real life example here from lambs from 2020. So we took Ivy. Now her phenotype, the color she is black. Now she's a catmugget, and we'll talk about the catmugget in a little bit and what that does to a black-based sheep. But for the purposes of this, she is a black-based ewe. So her color is black, so that's the capital B. And then Thor is the ram we bred her to, and his phenotype is also black. So we've got two sheep that have the black phenotype, but they had a brown lamb. So when you look at this, let's just pretend we didn't know what the dam or the sire carried. We knew that we were going to have a 75% chance of getting a black-based lamb. But because we got a brown-based lamb, now what we know from the sire and the dam, what we've learned is that they both carry brown. So now we know that for future breedings when we use those two, and if we want to either limit or increase our brown sheep in the flock going forward, we'll have that information about those two sheep. So here's another real-life example, and this time we're going to talk about spotting. So the sire in this case was Rush, and Rush, his phenotype is spotted. You can see that he's got spots. He's got the big spot on his neck, and he's got a white spot on his head. So actually the white part is the spot, which is kind of not counterintuitive in this case, but at any rate, so his phenotype is spotted. And then we have Arya, who was the dam, and she was not spotted, so she did not exhibit spots. But she could be a carrier for spots, so that was kind of what this experiment was. So we don't know what her second allele is. We know that she's not spotted, but does she carry spots? That was the question we were trying to explore here. Obviously, Rush is homozygous for spots. You would only be spotted. You'd only have a phenotype of spotted if you have both recessive genes for spotting. And look at what we had. We had this pretty non-spotted ewe. So that didn't really help us at all as it relates to does Arya carry spots or not, right? So we'll keep working on this. The thing about Arya's lamb, though, is that she certainly will be a spot carrier because she got the recessive genes from her sire, Rush. So pattern is a little bit more complex. It's the third and last one we're going to talk about. With pattern, there are 18 possible alleles in sheep, and different breeds of sheep have identified different numbers of those alleles. So I don't know for sure, but I doubt that every breed of sheep has all 18 alleles in their standard. But what I'm going to be talking about today as it relates to the Shetlands are just 5 patterns to sort of keep it simple. There is a hierarchy of dominance for patterns, so they're not all likely to occur at the same time. So there will be dominance within the 18, and it is actually a hierarchy. And there are alleles that are codominant, so that they can all present when they're found in the sheep. So the 5 patterns that we're going to be talking about in order of dominance are white, and that's the allele for that is AWT, tan gray, which is AG, gullmugget AT, catmugget AB, and then no pattern or solid. So one thing you notice with patterns, the allele is represented by more than just one letter. In the color and the spotting, we had one letter for each allele, and because patterns, there's so many of them, they require more than one character. So just don't be confused by that. I had a hard time with that when we first started doing this. So let's just talk about white first. White dominates everything. Nothing else will show. If a sheep has a white allele, it will cover everything. So it's tricky with the white sheep. You don't, unless you know their pedigree, it's difficult to know for sure what they're carrying. So when you see a white sheep, it could be black or brown based. Does not show because the white covers it. Spots don't show. Whether it's a gullmugget or a catmugget doesn't show. And if it's AG, that also won't show. Now there's work being done to see if there's any tricks or clues for if you can see certain characteristics or traits that maybe indicate that they're covering some of these patterns, but or traits, I should say, excuse me. But for the purposes of this, white is the dominant and covers everything. And the other thing about that, it makes sense, is when you think about sheep, you typically think about white sheep. So just it is logical. The next trait or pattern, excuse me, is the AG pattern. And what AG does is it makes solid colors lighter. So AG combined with a black-based hue can make gray, and AG combined with the brown-based hue can make musket. Fawn is actually not correct. I need to take that out of there. So only white is more dominant than AG. When it's mixed, or when it's AG and a color-based black or brown, those colors get lighter. Spots will show. Gull mugget or cat mugget may or may not show. I actually just read something this morning that indicated that I think they might be codominant. There's actually two AGs, gray and tan, but for the purposes of this presentation, I've combined them, which is fairly harmless. The next pattern is cat mugget, indicated by AB. Cat muggets are known by having a badger face, which is the bold eyebrow pattern. They're dark on the bottom of their body. The wool is darker on the bottom and lighter on the top. And then cat mugget is codominant with gull mugget. So for dominance, like I said, it's codominant with gull mugget. If the sheep is black or brown based, the color will change with the cat mugget. So black based sheep will end up being called gray cat muggets, and brown based sheep will be called fawn cat muggets. The spots will show in a cat mugget, and then AG may or may not show. There's additional patterns if you want to do, go into that deep end of the pool. There are additional patterns that are clarifying the type of cat mugget, which I'm not going to pursue that in this presentation. And then gull mugget is similar to cat mugget. It's kind of the reverse, where they are darker on the top and lighter on the bottom, and they have a little arrow-shaped pattern in the corner of their eyes. And then it's quite a fetching pattern. These are really cute lambs in that picture. Very similar to the cat mugget, so I won't go through the table. And then, yes, there are additional patterns that clarify what type of gull mugget you would have. And then there's the pattern called no pattern, also called solid, but technically when you read the documentation, they say lack of pattern. It is the least dominant of the patterns. If they are no pattern, the color will come through in its darkest form. So black-based sheep will look black if they're no pattern, and brown-based sheep will look brown, and so it's called morit. Spots will show. If they have an allele for gull mugget or cat mugget, that will cover the no pattern, and AG will also cover no pattern. All right, so let's do a real-life example of patterning. So we have ewe Ophabia, who we bred, and her pattern phenotype is white. And we bred her to John Snow, and his pattern phenotype is cat mugget. So he is homozygous for cat mugget. And in this scenario, let's pretend we don't know what the Ophabia carries. So that's where we leave that with a question mark. So when we take a look at what she had for lambs, she had 2 white lambs. They're showing a pattern phenotype of white. So in the end, that really didn't help us to understand what we have with Ophabia. But we do know that the lambs will carry the cat mugget pattern. We actually think Ophabia is homozygous for white, but you never know. Well, you might get surprised. Okay, another real life example. This time we're going to look at Circe. So she's got a pattern of cat mugget, and we bred her to Rush, whose pattern is no pattern. So that's kind of confusing, right? Because he's got spots, so you would think he's got a spotting pattern, but spots are a separate location on the chromosome. They're a different set of genes, not related to patterning. So when you look at Rush, he is actually no pattern. Solid, the very bottom of the dominance hierarchy. And because it's the most recessive, he's homozygous for that trait. And Circe is a catmugget, but I'm not sure if she's a pure catmugget or if she is carrying something else. So let's take a look and see what they had for a lamb. They had a beautiful black ram, and he is solid. No pattern. So we did learn something here. We learned that Circe carries the no pattern or the solid phenotype gene. So that's interesting information that we learned from that breeding pair. So I want to talk a little bit about the color wheel. You know, I've always had a few questions about this color wheel. So what I wanted to do was sort of lay out where I am right now with how I'm organizing the colors in my head as it relates to the genetic makeup that creates that color. So I think that if you look at this poster, you might be under the mistaken perception that we just look at the color and look at our sheep and say, yep, looks like it's Emskit, or, you know, yep, it looks like it's fawn, where actually those colors are an expression of very specific patterns and color bases. And so you have to know the genetics of your sheep before you can identify them as, let's say, Emskit or Shayla, because it relates, the color on that table, the names of the colors relate to specific patterns. So for example, Musket, that is and Musket is maybe 7th on the list here. It's a brown based color, so your sheep is going to be, if it's Musket, your brown genes are going to be lowercase b, your color genes. And then it's going to have an AG pattern, which lightens it to Musket. So that's the only time you would call your sheep Musket. One thing I'm not clear on is where do the catmuggets fit in here? So that's something I'm still trying to figure out. We didn't talk about the modifying gene. That's another gene location on the chromosome that changes the color ever so slightly. I didn't get into that particular one. It's not a pattern. That's actually its own trait that affects the color of the fleece. So I just wanted to sort of point that out, that you know, it's not just as straightforward. There's a lot of work underneath before you can select the color that you register your sheep in the registry. And that kind of takes us to, you know, I'm still learning. There's so much to learn, and people are, you know, really working very diligently to try to figure out how to define all of these, you know, the genetic makeup of the animal, what makes them a Shetland, right? And so there's other types of genes like extension genes, the modifiers that we talked about that, you know, impact the color of the wool. One thing too that I'm interested in understanding more is with spotting. So there's a lot of different types of spotting, right? You've seen in my flock the euglet with the panda eyes and the ones that have like the white stripe down the front of their face. You know, Rush, who's got kind of this palomino type of spotting on his body. So how do you control for those kinds of things? Horns are also, you know, relying on genetic makeup for what is expressed in the animal. You could have Shetland rams that have really impressive big horns, or you could have rams that are polled with nothing. You can have horned ewes as well. Those are acceptable in the standard, but that's another whole genetic puzzle to understand. And then, of course, those 13 other patterns. How many of those apply to Shetland? Again, these are interesting questions that people are working on trying to understand today. So hopefully that gives you a real simple overview of the pretty much the key genetic things that we focus on when we're breeding, and hopefully it gives you an idea as I'm talking about stuff going forward in my videos. You know, you can kind of have a better feel for what I'm talking about when I throw out these terms. Any questions or comments, feel free to leave those below. And if you found this helpful or liked it, you know, please give me a like, and I really would appreciate that. Thank you very much.